Oh, welcome everyone to the Precision Medicine seminar series. It is my pleasure today to introduce John Novembre. Of course, I mispronounced your name. Um, he is a professor in the Department of Human Genetics and Ecology and Evolution at the University of Chicago. John is a population geneticist. He is very much interested in computational methods to address human diversity. And these methods are broadly applied to understand the genetic patterns of variation across different geographic regions. He recently served on the National Academies of Science, Engineering, and Medicine Committee on the use of race, ethnicity, ancestry as population descriptors. And I think there was a pretty good Columbia representation also on that committee from what I saw. His work is supported by the NIH funding. He is also recognized as MacArthur Fellow, Searle Scholar, and Sloan Research Fellow. Multiple publications. He's also serving as an editor of the journal Genetics. And we are very lucky to have him today to tell us a little bit about how to define population for genetic studies, and he will talk about genetic study design, a topic I think that's relevant to many of us here in the room and on Zoom. Thank you.
So much. All right. Thank you for your introduction. Thank you. Great. Well, yeah, it's a pleasure to be here. I want to start with acknowledgments. I want to acknowledge that the main project I'm going to talk about is work by Maggie Steiner, Dan Rice, Marita, Yanni Rowan, Arjun Badana, and Chris Forrest. Current graduate students, these two, former postdoc, former student, former undergrad who's now at Sinai, actually, so in the neighborhood here. And so I want to begin with some background on my lab and its research interests and some ongoing projects. So that'll be sort of the part of the talk where I get to sort of give you an idea of some of the stuff that's active and ongoing. And then I'll get into a deeper story about sort of the topic that Christophe just mentioned on sampling design and thinking about the scale of sampling and populations in our work. And great. So broadly, by way of introducing my lab and its interests, I sometimes like to use this diagram here of the tools that are sort of what we use day to day. And this is sort of what we specialize on a technical level. So the tools of computational statistics and theoretical population genetics, that's what we're training in. And then we're using these tool sets to address topics and kind of the intersection of evolutionary genetics and human genetics and genomics. We're kind of swinging between those two. And more specifically, a lot of our contributions have been in the space of thinking about like conceptually, methodologically, or developing resources that help us understand the structures really of genetic variation and the history of genetic variation. And with a special attention to geographic structure and also haplotype structure. So being more specific on that, one theme in my lab has been understanding and applying low-dimensional representations of genetic structure. So we had some papers in the arena of understanding how PCA behaves with spatial data, applying it, doing admixture type estimation. And these days, what we're up to, the section about is briefly, is just I'm going to hat tip, several active students in my lab and their projects. So Ahmed Nuria, who's a visiting student from Barcelona, from UPF, and Luke, a current postdoc, have been doing work on a kind of PCA type approach, low-dimensional representation that's stratified by time. And so you can see not just that there were two populations here, but that at some point in the past, they actually were sort of more close to each other than they are right now. They were exchanging migrants, and at some deeper time in the past, they were actually like perfectly well mixed. So that's a sort of ongoing project in that arena. Another big area for us over the years has been developing migration maps as a tool for understanding genetic structures. We've had a pile of papers in this arena, gradually like making these maps better in terms of their speed of computation, their what they're capable of representing. And some of the most recent stuff with Viv is we have these massive migration where there are like regions of low estimated gene flow and high estimated gene flow, like in blue, that represent how genetic diversity is structured on the landscape. So you can actually see that there's more genetic differentiation across the Mediterranean or across the Sahara than like along the edge of it. And we've now been adding in the ability to recognize when there are patterns of genetic similarity that are kind of represent long distance relationships. So Madagascar is a really interesting one. That like in the basic version of these maps, we couldn't fit Madagascar very well because Madagascar has a large portion of its genetic ancestry coming from Southeast Asia. Actually, the peoples of Southeast Asia moved, the inferences by sail to here and founded a lot of the played a large number of the genetic ancestors of Madagascar people trace back to Southeast Asia. This is now like inferable kind of automatically in the software through this this added feature that we've put in. And it detects a number of other like interesting population movements that we kind of need to add to the model to have it be a good reflection of the genetic diversity that's there. So these are an alternative to the kind of low dimensional representations that I showed, using the spatial information, building these maps. And then another extension to these that Hao Shen, a postdoc in my lab, is working on is this the pointer. Oh, the pointer on the screen, is that? Yeah.
I'm sorry, John, also the Zoom people.
And the Zoom can't.
Maybe the volume is a little bit low. So
oh, yeah, I should stay closer. Yeah, I'll point with the mouse. I'll stay closer to the, okay, so, and then let's see. Great. So, all right. So then Hao, a postdoc in my lab, is working on versions of these maps where the edges are directed so that you can infer not just that a region has sort of is best explained with a high or low migration level, but with a directional migration. And so this map is actually inferred using ancient DNA from the Bronze Age and Iron Age. And these arrows, like particularly in this region, they're kind of pointing from west to east, reflect that a lot of the genetic ancestors of these regions came from the east, which is in line with a lot of ancient DNA studies arguing for the expansion of peoples from the steppe, the Pontic-Caspian steppe, sort of this region, westward into Europe. And so this software is kind of allowing us to study those types of movements, but in a very agnostic way. We're not sort of doing specific tests. We're just sort of feeding the data in and seeing what kind of migration rates and their directions emerge. Okay. So low dimensional representations, these effective migration maps. Just broadly, another theme in the lab has been like resources and studies of human genetic variation. So a bunch of different studies in that arena, some focusing on the Sardinian population, some focusing on Mexican biobank, some this was a large study of its time, 14,000 people, 202 drug genes. So I'm going to come back to this one in a minute. Developing a browser that allows you to look up variants and see the allele frequencies across the globe. Kind of a very straightforward, but very turns out useful reference for many people. And we've been extending this browser to try to show LD levels of variation in LD across populations. And then another version of the browser that operates in a low dimensional PCA space that sort of allows you to do matching, like learn what allele frequencies are at particular locations in like a PCA space versus geography, or versus using a broad population label like European or African, given some of the challenges with those types of labels, given they're not very specific. Okay. Another running theme in the lab has been looking at positive selection in humans. And so a couple of different studies in that arena. Again, two of these coming out of the collaborations in Sardinia. One going back to my PhD work even. But most recently, we've been developing an algorithm that allows us to use ancient DNA time series to study selection. The challenge there is that there's a lot of migration in the history of human populations, and you worry that perhaps what looks like the increase in frequency of an allele due to selection could actually be another population moving in. And so Shinyi's developed a method that kind of, if I focus right here, in the case where you'd have the strongest biases from a source population with a high initial allele frequency that's different than what you started with, her method is the one in orange here that's giving close to recovering the true values, whereas other methods are either biased or have large standard errors. So that's a space that we're working in. Okay, so those are a lot of themes of things that like you may have seen come out of my lab, but giving you kind of a bit of the latest. This is a rather new area, at least in the direction we're going. So we get stratification in GWAS studies. So there's been a little bit of this in my past in terms of like in this genes mirror geography within Europe paper. We looked at how does phenotypes that vary by longitude and latitude lead to inflation of GWAS. Here, what we're looking at is how do multi-generational shared environment effects, think about like things that run in families over many generations, and how do they affect outcomes like this, phenotypic covariance versus genetic relatedness. These are the outcomes that are used to estimate heritability in Haseman-Elston estimates of heritability, where the slope of this line reflects how heritable a trait is. And in some large biobank scale studies of phenotypic covariance versus genetic relatedness, there's been the observation that the slope of these in this relationship is oftentimes nonlinear, with like these kind of bumps that occur in the relationship. And we've been exploring how shared environment might explain these bumps, where and sort of the preliminary view is that as you traverse different levels of genetic relatedness, you're passing through different levels of pedigree relationship in terms of first cousins, second cousins, third, fourth, fifth cousins. And each of those degrees has different levels of shared environment. And so you're getting some of these more punctate drops in the slope around where you're shifting between these relatedness categories. And we're bringing that up because the alternative hypotheses for this involve like LD relationships or complex arguments about epistasis, but we're bringing up the kind of playing out like how can this work if it's shared environments that might be explaining these phenomena. So that's with Marita, a student in the lab. And then not any particular student, but me myself, I've been had a theme of trying to have more reflection and give perspectives on the field. And so there've been a couple of different contributions in this area, but most recently, as I mentioned, this NASEM report has been something that we've been that I worked on extensively, and so they're quite an amazing multidisciplinary group that came together to reflect on the use of population descriptors in genetics and genomics research. And maybe one of the key things is asking everyone to be more reflective about how they use population descriptors. Ask themselves, why exactly are they using population descriptors in their study? What's the work that population descriptors are doing for you? And ask if it's the right work for the question you're asking. And so this table, that's one of the major outcomes, is for each study type and each of different possible categories of descriptor, race, ethnicity, language, geography, genetic ancestry, genetic similarity, whether that category of descriptor is actually what makes sense to use, or is maybe like a proceed with caution type issue. Okay. So I'm going to use that to segue to today's talk. Okay. So one of the things that came up in the report was thinking about terms like European genetic ancestry. It's a continental ancestry term, right? And we say many GWASs occur in European genetic ancestry. I did my GWAS in European genetic ancestry. But yeah, what is European genetic ancestry? What is our study population here? There are many possible scales at which we could do this, right? We could have a study that is just based on the medical systems around Manchester, Sheffield, or Stockport, which turns out to be where the pilot study for the UK Biobank was carried out, right? We could go across the UK, looking at like, these are all the study centers of the UK Biobank, or we could have samples from across all of Europe, right? So I want us to be thinking about scale, spatial scale of our sampling, and how it impacts the type of variants we discover. And so I'll stretch this beyond like European genetic ancestry to the scale of the whole globe. And I'm going to be contrasting in this talk narrow focus sampling, where you get all of your individuals from sort of one geographic location, versus broad sampling, like sampling, say, across the globe. And a key tension is going to be like for like a fixed sequencing budget, what are the trade-offs? We're going to think about like lots of individuals from one location or fewer individuals from a broader set, okay? I'm using geography to frame this, and you can think about that in terms of geographic ancestry, like individuals who have their ancestors, their genetic ancestors from one geographic region, as sort of the sense of geography that we mean here, okay? But you can also think about this problem like in a genetic ancestry space of like, are we focusing just on one corner of the PCA space or broadly across, a big swath of it, okay? Okay, so yeah, how does sample breadth impact our discoveries? But another way, what is the impact of sample design breadth on variant discovery? Okay, and here's this team that's been working on this that I've already introduced. Part of our motivation is the study design in the biobank era. So we've got many different biobanks forming, and we could think about, if we had a big bolus of money, should we spread it out across all of the biobanks and helping them top up their sample sizes, or should we pick one and go really deep in that one biobank? And so that's a very active area of current research. Another motivation is thinking about genomics for the world. So this very influential piece by Carlos Bustamante, Esteban Burchard, and Francisco de la Vega, in really pointedly calling out the focus on European descent individuals in medical genomics. What was this? 5 years later, Genomics is Failing on Diversity sort of still, by Alice Popejoy and Stephanie Fullerton, pointing out that there had been some change, but still at that stage, a lot of studies are still focused on European ancestry. So that's that tension about what is the impact of sample design in terms of variant discovery is sort of is how we want to think about it here. Okay. I'm also going to frame this problem with a big interest in rare variants. Okay, so this is a figure from that study that I mentioned that at the time was a large sample size. It was one of the first studies that had sequenced a subset of coding genes in a large number of individuals, sort of getting over the 10,000 individuals level. And this was a plot that we made in that paper. The number of variants greater than 0.5%, the number of variants less than 0.5%. And this with the kind of punchline of this figure being common variants being the tip of the iceberg. The things that you see common in your gene are a small number relative to the number of rare variants that you discover as your sample size gets quite large, okay? And these rare variants oftentimes have large effects. So from this Maruli et al. Paper in 2017, and now there are many, many such graphs, but this was for height, and you're seeing the effect size as a function of the frequency. A lot of these rare variants have large effects. One of the major ideas in the field right now is how these big effects might give us more actionable insights. Okay, this quote from Suzatovsky et al. Right, that these rare private functional genetic variants, the highly penetrant gains or loss of functions, give us the most actionable insights. Okay, sometimes there's another quote in that paper that rare coding variants jumpstart biological hypotheses. Okay, and if you think about like the omnigenic model that Pritchard's put forward, this idea that the rare variants might be perturbing core pathways relevant to the phenotypic outcome, as opposed to being like slight perturbations of distal gene regulatory networks. More, in that line, this beautiful paper by Barton et al. In Nature Genetics showing for various phenotypes and these particular genes, large effect variants that cluster within particular genes, and look at these minor allele frequencies shown in these side panels, 10 to the -4, 10 to the -5, or 6 there. So extremely rare variants with large effect sizes clustering within particular proteins. Okay, and these rare variants, they are often geographically clustered, and I'm gonna dwell on that point. But this is from a paper that I was involved in some time ago, looking at LRRK2, which is involved in Parkinson's, and in particular looking at a number of variants relevant to Parkinson's, and one of them in particular is R1628P. So the pluses here. This is the location of carriers of this mutation within a broader plot of genetic ancestry, we could say genetic similarity on a PCA plot, representation of the genetic similarity of these individuals to each other, and the carriers of this rare coding variant are sort of clustered in that space. Okay. So that's going to be something that's an important backdrop for the type of thinking that we're going to do. Okay, so more backdrop is reflecting, and it's kind of shown in this figure, that the human genetic variation is really a continuum, and so and this is quite well. This is from the BioMe biobank cohort from Sinai right here in New York City, right? So it's PCA showing this continuity. This is from this older work I was involved in, looking at genetic diversity in European descendant individuals. But this history that we have of arising in Africa, a subset of humans moving out across Africa, and kind of smearing variants across the globe is really essential to understand. And so we'll be keeping that in mind as we try to build a mathematical model of to help us understand this question of sampling scale. And another kind of backdrop is what we learned from looking at a lot of variants. Okay, so what I'm showing here are two figures from this work that we did on trying to create visualizations that help people understand human genetic structure from a variant perspective. Because what we found is that maybe a lot of students in human genetics have seen thousands of PCA plots or dozens of PCA plots and maybe admixture type representations of human variation. But if you ask them, like, what is a typical human variant distributed like, they might struggle to actually be able to describe that, to say, oh yeah, there are a lot, like if you said, are there lots of variants that are, is there a substantial fraction of the genome that are variants that are high frequency, let's say greater than 80% in one region of the world and absent everywhere else? Okay, it might struggle to actually, answer that. So, so these two figures kind of get in that arena. So the one on the left is an example of 100 variants that are, each row is a variant, and each column is one of the samples taken by the 1,000 Genomes Project. So Urubans of Ibadan, Tuscans of Italy, et cetera, okay? And these are organized left to right in broad sort of geographical arrangement. And the colors represent the minor allele frequencies. And when you see of these 100 variants, Most of them are rare. That's that sort of tip, the common variants are the tip of the iceberg type. We see that again. There are very few variants that are kind of common, but also we notice an interesting kind of partition, so to speak, of lots of variants that are rare and found in just one location or like two neighboring locations. Okay. And then a bunch of variants that are common and found everywhere. And then, some middle ground of variants that are like a little bit of an intermediate frequency and shared amongst their geographical neighbors. Okay. This is a more thorough, extensive version of this, where we actually classify all the 90, roughly 2 million variants that are found in the New York Genome Center's resequencing of the 1,000 genomes data. And so the 3.1% sites of the whole genome that are of the measurable genome that are variable, those break down into categories that we show here. And the way to read these categories, it's meant to be like fairly approachable. An R means that the variant is rare in the African supergroup, like population set of populations in the 1,000 genomes. And then U means undetected everywhere else. This is rare in East Asia, undetected everywhere else. Rare in South Asia, undetected everywhere else. Rare in Americas, undetected everywhere else. Rare in Africa and the Americas, undetected everywhere else. Rare in Europe, that's undetected everywhere else. And then common everywhere, okay? And then more like rare and shared between populations that have shared genetic ancestors. And then this is common in Africa, absent outside, except shared with the Americas, okay? This is really a snapshot of what variation in humans looks like, the 1,000 genomes data set in particular, and it should resonate with our understanding of human population history. What's going on here is you have a species that was together in Africa, variants that were common there got spread out across the whole globe. So things were common in Africa, spread out across the globe in general, sometimes lost. That's what you're seeing here and here, okay? And then after spreading out across the globe, human population sizes, they're growing, they're increasing, a lot of new mutations are occurring, and they're in little pockets in different corners of the globe. They're spreading, but it takes time to spread. It takes time for their numbers to increase. Crucially, there hasn't been enough time for variants that are to increase to high frequency locally and be absent everywhere else. Okay, that's happening in a couple cases due to positive selection, driving things up quickly. But like your average thing that has very weak effects on fitness is localized. Okay, and then especially stuff that's deleterious, that's damaging human biology, right? That's having fitness consequences, is going to arise, have a short life, and die. So it's also very geographically localized. So we're interested in, I think, or at least I'm going to focus this talk in terms of thinking about rare variants that might have a large effect on fitness, large effects on human biology. The idea is they're going to be some of these variants that are kind of very geographically localized. So what is the consequence of spreading out our samples or concentrating them in detecting these types of variants? That's the question. Okay. There's been relevant previous work. Yeah. Essentially, yeah. We're basically
exploring much the variants that are very common multiple population and the absent in some.
Yeah, what's going to happen is that I'm going to develop a mathematical model. The mathematical model is going to make approximations about things being rare, and yeah, so it's going to be most applicable to the rare stuff. I think the broad insights, some of them like carry forward as you go a little bit higher frequency, but that's the realm in which I mean, yeah. Yeah, okay. Okay, so then, right, so there's some relevant previous work, okay, and in the context of looking at neutral variance. Okay, so largely driven by an interest in understanding how we can reconstruct the past. The abundance of rare variants versus common variants can tell you something about whether a population has been growing or not, or whether it's been through bottlenecks. So a number of the geneticists who are interested in understanding the human past or the past of other species ask themselves, well, if we sample just locally versus broadly, how much is that impacting our discovery of these different categories of variants? Because that's going to impact how we reconstruct the past. And in those cases, they found that local sampling tends to increase the relative number of intermediate frequency variants you get over rare ones, okay? So you have studies that were done kind of like empirically by resampling human data in different ways, theoretical studies, simulation studies, empirical studies, simulation studies, empirical studies, okay? This is kind of through a little time trajectory. Yeah, in general, motivated by reconstructing demographic history. This Gloss et al. Paper is slightly different. They're interested in complex trait mapping in Arabidopsis. So this one's quite kind of the closest empirical precedent to our study. And fortuitously, we connected with Andy because he was working with Joy Bergelson, who was at UChicago previously. And there's some nice kind of interesting parallels here. And we've written a perspective piece together actually on it. So these previous works, though, they have this focus on neutral variation. Our interest is deleterious variation, these things that might be perturbing human biology, having fitness consequences, okay? We also are interested in much larger sample sizes. Most of these studies were done thinking about sample sizes in the tens to hundreds. We're interested in like biobank scale, okay? So, and we're also interested in the consequences of this for phenotypic associations, okay? Now, the possible approaches we could take to work on this could involve using simulation, okay? But that's going to be very slow for biobank scale studies, okay? We could do empirical analyses by like resampling the data, and we're going to do some of that. But it's maybe not fully pleasing because we can resample the data, but we don't actually know like what variants are the most, actually do have these like fitness consequences or do have these phenotypic effects. And we could do theory. So the theory is challenging because it requires modeling many different aspects at the same time. And population geneticists are generally good at doing like 2 or 3 of these things at a time, like mutation and population size change, or mutation and geography, or selection and mutation. But this has mutation, geography, selection, and then also non-random sampling, okay, where like you're going to concentrate your sample in one area or spread it out uniformly, okay? So we're going to start with the theoretical approach here, okay? And we're going to take inspiration from, this is John Hart, who was a professor at UC Berkeley, where I did my PhD. He taught this popular course. It was called Consider a Spherical Cow, a course in environmental problem solving. This is kind of the reference here is like you're trying to understand like something like how do cows affect like the global carbon, so you need to think about how much grass is going into a cow, how much is a cow, emitting in terms of biomass, et cetera. And of course, maybe the thing you want to do is just imagine each cow is a little circle with an arrow going in and an arrow going out. And so we're going to do something like that for human biology here. And we're going to sketch out this problem in a very simplistic way. So we're going to have sort of a sense of geographic space that's continuous, okay, inspired by that continuous nature of human variation. We're going to imagine mutations arising equally likely at every point in this space. These colored clusters are meant to represent different carriers of a single mutation. And then we're going to imagine the sampling in a very abstract way as just being like from a Gaussian distribution that either has a large scale parameter or a narrow one, Okay, and at the limit, it'll be like uniform sampling. But and another limit will be point sampling, and then we can have kind of everything in between by varying the scale of this Gaussian. Okay, and so the idea is like our broad sampling will discover maybe 3 carriers of this set, each from a different kind of cluster, or the narrow sampling might discover like maybe multiple carriers from a different set of clusters. Okay. So that's how we're conceptualizing the problem at first, pass. And then let's say, okay, let's build a mathematical model. And so what I'm going to show is the mathematical model has a notion of time in it. So I'm going to switch to like a one-dimensional space and a two-dimensional time, or sorry, one-dimensional space and then show time on the x-axis. And now each of these diamonds represents the first mutation, the first time, like a loss of function variant occurred. And then this is its sort of lifespan as it passes down through different offspring, and it's carried by multiple carriers. And then those carriers die out and they're spreading geographically over time. Okay. So this is a kind of spatial birth-death process. And we imagine at some point, like sampling the population, and we can either sample like a narrow area where like the width here is this width, the scale variable W is 50, or sampling a broader area, 100, but with the same like sampling effort. Okay. And so we will just have different discoveries as a result of that different sampling scale. Okay, so this model then has mutation rates of how often are these mutations arising, how long are they persisting, and that's affected by selection. What's the density of the population? Because you need that times the mutation rate will determine actually the total rate at which new mutations enter. There's a dispersal parameter, so they're dispersing according to a Brownian motion. So they're sort of like taking random walks in space. And there's a scale to that that's about like related to the scale of dispersal generation over generation, how far offspring moving from their parents. And then there's that W, that scale of the sampling itself. Okay, so those are all the different players involved here. Okay, any questions at this point? Okay, okay. To set up this scale variable, this is very like spherical cow type stuff here. Okay, it's the distance in the model is measured in terms of sigma. Okay, so and this mean square parent offspring dispersal distance. Okay, the scale at which parent offspring disperse from the average of their parents or the parent in a haploid model, and so estimates of sigmas in humans vary from 1 to 100 kilometers, and it depends on where you are in geography and where you are in time. But if we assume that it's about 10 kilometers, then for sigma, then in those units, the width of Europe is about 400 sigma. The longest land path from Africa to Eurasia, shown here, is 1600 sigma. The circumference of the Earth is 4000 sigma. Okay, so then W values, which are going to be measured in sigma, so like a W value of 10 sigma would be something a little bit less than the width of Europe. Okay, something that is 10, like 100 or larger, is going to be more like the width of Europe, or getting to the scale of Africa and Eurasia. Okay, so like 10 you can think of as kind of local-ish, like a subset of Europe. 100 or larger, I think they're starting to get to global scale. Okay, all right. Okay, so we're interested in understanding the site frequency spectrum. Okay, this is really kind of like a vector valued object in population genetics. We have the minor allele frequency of different variants, or minor allele count equivalently, kind of on the x-axis, and the number of variants per kb that have that particular frequency or that particular count. Okay, so this would be to say, this is from that sequencing of drug targets study. The number of singletons per kb in that study was a little, somewhat over 10. Okay, the number of 10 tons was something like 1 per kb, and so on and so forth. And the site frequency spectrum in constant size populations has this nice result that it should have on a log plot, should have a slope of -1 for neutral variance. And then for deleterious variance, it kind of breaks off from that curve because selection is preventing the variance from reaching these higher frequencies. The neutral variance can get up to these higher frequencies, but the deleterious ones are pushed down. They're kept to the left here, okay? So, and you can kind of see this in this actual human data where this blue line are the non-synonymous variants, and you see this gap here. There are fewer of them than we'd expect compared to like your synonymous variants or UTRs or intron variants, because selection is presumably pushing them down and out, okay? So we're going to be trying to solve for this site frequency spectrum as a function of W, okay? And the site frequency spectrum is great because it allows us to learn things like the total number of variant sites that we would discover, also called the number of segregating sites, which you might see on some of these plots, the expected number of singletons that we'll see, the average frequency of the variant sites, the average heterozygosity, things that are super relevant for our power in mapping studies, okay? And to do this work, basically, we set up a system of equations that describes the moments of the sample frequencies, and we can use those to get to the SFS. And this moment generating functional that will allow us to get the moments can be written in terms of this function U, which itself we can solve using a system of a partial differential equation, okay? And this isn't a math talk, but let's just say there's a lot of kind of like applied math and tricks to sort of working with these equations that allow us to make progress. And one of the things you find when you spend time with this system is that you can boil it down and actually see that it's mainly driven by 2 parameters, what we'll call an effective mutation supply and an effective selection intensity, that they themselves depend on W, on the sampling width, okay? So we're going to present results in terms of these 2 parameters, but let me explain like how they work. So if this effective mutation supply is large, then it increases the intercept of the site frequency spectrum on the y-axis. It's like saying there's more mutations, so there's like going to be more singletons coming in on the left side of this graph. The effective selection intensity is about how quickly does that site frequency spectrum get cut off? Like how shifted to the left are the variants, or how much are they prevented from going to the right, okay? So increasing gamma E, the effective selection intensity, it decreases the number of intermediate frequency variants. The stronger this parameter is, the more this line will break and go downward sooner, okay? More like, and if let's think about this distribution, we're shifting it left, that means we have lower average frequencies, okay? So some results from theory, okay? So these are the effective mutation supply and effective selection intensity as a function of this width parameter. As you make a sample wider, it's as if you had more mutations. You'll discuss the y-intercept will go up, okay? Also, as you sample more widely, the gamma E parameter goes up. So you're also cutting things off, and they're sort of, they're not reaching as high of a frequency, okay? We call this upward shift in the SFS the discovery effect, that you're going to see more variance. And then this gamma E effect, called the dilution effect, that like as you make your sample more broad, the variants you find, they're going to be kind of diluted out in a background of non-carriers. And so their frequencies are going to be lower on average. Okay, so let's see this. These are site frequency spectra now shown for different choices of W. And we're getting these results for biobank scale samples, 500,000 here, for negatively selected variants that are strongly negatively selected. And here's what we see. So when W is narrow, like 1 or 10, you have a smaller gamma E, and so you can see these variants at higher frequencies. And we have a lower theta E, so there's a lower y-intercept. And you can follow that through to looking at broad samples where we have more discovery of singletons and doubletons, but a quicker drop off. They tend to be at lower counts. Okay. And this actually aligns with the kind of cartoon here that in broad sampling, you're going to see more variants, but they're each at a singleton count here, as opposed to the narrow sampling, we saw only a few variants, but they're kind of concentrated. We get to see them at higher counts. Okay, they're more shifted rightward here on the count distribution. Okay. We checked our theory against simulations that don't make some of the approximations that we needed to make to get these theoretical results. And these are comparisons of the theory and the simulation. So this is just to say that like the math is checking out. What's nice is these simulations take about 8 hours to make 50 replicates to get like these dots on the graph here. The math takes about 9 milliseconds to solve these equations once you have it all set up correctly. Okay, so that allows us then to explore a lot of choices about W and S and N, and so this is sort of a set of results exploring over that space. And let's see, the columns here are the proportion of segregating sites, the proportion of singletons, the segregating allele frequency, and the heterozygosity. The top row has a fixed sample size and varies the selection coefficient. The bottom row has a fixed selection coefficient and varies the population size. Okay. And we see in general, like I won't dwell too much on the subtleties here, but as the sampling width gets larger, you get more segregating sites, more of them are singletons. The frequencies, though, at which you detect these variant sites goes lower and lower. Okay. And then the heterozygosity is actually pretty invariant. It changes with the selection coefficient, but it's not variable with the sampling kernel. And that's kind of a surprise and interesting result. Okay. And then as a function of sample size, you can see these slopes get more steep. So, and the range of where the effects of sampling width arise are kind of broader. So all of these effects, when you get to biobank scale, are kind of larger, right? That the sampling width is more important as you get to these really large sample sizes. Okay. So while this may seem really abstract, I want you to think about like what drives power for you in your GWAS study. It's the number of times you've seen a variant, right? So this segregating allele frequency, might be really important for, you've seen that allele. What are the types of numbers of times you're going to have seen that to make an association between carriers of that variant and a phenotype? Okay. What about like in a burden test? It's like the number of loss of function variants that you've seen total, regardless of exactly which locus it occurred at. So, the raw kind of numbers of segregating sites become important. So we've actually done some looking at this and kind of passing these site frequency spectra through like power curve calculations. And this is one of the preliminary results, is sort of as the sampling width increases, you have, for weak effect alleles, you have this huge penalty to power because you're getting things like washed out down to singleton type counts and you can't make any associations unless the effect sizes are really large, because when effect size is really large, then you can make a discovery even with a very small number, right? And then you're getting lots of them, so it starts to balance out. With burden tests, where you're collapsing all these, you don't mind that you got lower numbers per variant because you have lots of variants. And so you collapse that and you actually increase power as you sample more broadly. Okay. So that's what the theory tells us. Okay. Going beyond the spherical cows, okay, and this is we want to test some of these predictions looking at the UK Biobank, and this is kind of the reverse of this sort of the masochistic undergrads of UChicago. They have this these sweatshirts that say that's all well and good in practice, but how does it work in theory? Okay, like very theory oriented campus. And but here we've started with the theory, and now we're going to ask how does it work in practice? All right, and so this is a subsampling experiment that we've done with the UK Biobank. And this is, I'm going to show kind of yeah, I'm showing results that are from this particular version of the experiment. We have a new version that we're just about to finish up to finalize this for publication. But in this version, we take a sampling center, like a location in the center of the UK, and we take broader and broader samplings going out from there. Okay, always subsampling 10,000 individuals. Okay. And so what happens, 10,000 individuals chosen very different ways, what's going to happen to our discoveries? As we go to uniform sampling, we get more segregating sites from the closest to the most uniform. The number of singletons goes up, the allele frequencies goes down, the heterozygosity stay about the same. So the theory is really kind of guiding us well. And here we can start to get a sense of how much these drops or increases are, and get a sense of how that might impact power. Okay, we have a similar experiment with PCA space, sampling at different scales, picking a location in PCA space, and then going out further and further from there. Okay, and this is a summary of both of those sets of results. This is preliminary results. Like I said, we're doing some touch up on this before publication, but this is a flavor. So this is from the closest 10K, like get as close as you can to one sampling center. Okay, or spreading out uniformly over the space of Britain, or uniformly over the PCA space. Okay, and what you see are like numbers like 1.13, 1.22x larger number of segregating sites when you take a uniform sample versus a point mass sample or a dense sample. Frequencies that drop about 12%, like 0.88. Okay, so these are the types of fluctuations that you might think power for rare variants is largely driven by the frequency. So for a variant site, it's about what's its frequency. So you'll see these kinds of drops. Okay, but you're also getting more segregating sites. Interestingly, right, those are compensating each other in a way that the heterozygosity stays the same. Okay. But your GWAS power, is kind of really affected by this. Your burden test where you get, is getting affected by this mostly. Okay. So that's sort of a view of this empirically, and gives us a sense of how this might affect us in practice. Okay. So some key takeaways: going narrow but deep, you get many observations, same variance, and enhanced power for single variations. Going broad, you're going to be able to get many variants, but possibly, only at singleton or doubleton, tripleton type frequencies. So it's going to decrease power unless there's super large effect size variance. Okay. And then there's some caveats here. When we were thinking about broad sampling, we're not adjusting for like the fact that like the issues of stratification. At broad samples, you're exposed more to confounding. You need to do corrections for confounding, and that adds degrees of freedom that will affect your power. Also, this has really been focused on like variant discovery, the number of variants you see and what their frequencies are. But there are a lot of other dimensions to this problem. Okay, so laying some of these out. Some of the complicating factors is, spatially varying environments, right? That if you're make a narrow sample, you get to sample a group that might have similar environmental exposures, and that might help improve your power, right? Broad sampling, you have the confounding. You also might, if you have context-specific effects like G by E, and like it varies across different E's, what the effect is, you might average out some effects that you might see if you hold the environment constant. In terms of LD, if you go to a narrow sample, you actually should also expect higher levels of LD, and so that's going to improve your power. You go broad, you're going to decrease LD, but that's better for fine mapping. We all know that trade-off. When you go narrow, sometimes you can focus on bottleneck populations where they might have particularly damaging variants at higher frequencies. Think like, like the number of discoveries that have taken place in FinGen and in the Sardinia projects. But you also have a saturation that like at some point, you're just sampling the same genetic lineages and you're not actually discovering more variants. If you go broad, you're going to avoid that saturation issue. Then there's a question of for prediction, right? Narrow sampling, you might discover some rare variants and make some associations, and that might be perfectly fine for like doing precision medicine on that target population, but it won't port very well to others. Whereas a broad sampling, you might, obviously get better portability. And then there's the question of like equity and inclusion, which is a huge one here, right? If you go narrow, you're probably, because whatever you discover there's a question of how well it will port to other populations. If it's an insight about core biology, it likely will port, right? Like if it's some key mechanism that's shared in our universal biology, it doesn't matter that you discovered it by sampling very deeply in one part of the world, because the mechanism just needs to exist across the globe, right? But if you go broad, you're probably for like the prediction applications, maybe going to have better ability to do prediction, and there's just the inclusiveness of actually involving all groups, right? So so those are some added wrinkles to this. I should finish up. So there's another dimension of this, which is for evolutionary genetic studies. So evolutionary geneticists actually use the observation of how does this shape look? Where does this the frequency spectra break in terms of like dropping off, and as a representation of how deleterious variants are, to infer how deleterious they are. But here what we see is where it breaks is strongly dependent on how you sample. And in particular, in some evolutionary studies, they go to one location and sample, a species, and then try to, do work on saying this is what the, negative selection coefficients are, and that's in that group. And the implication here is that like that type of approach would underestimate negative selection, because you focused on one area, you're seeing the negative, you're getting like elevated frequencies of the deleterious stuff right there. And these kinds of graphs here indicate that like to get something that's reflective of the true gamma at the level of the whole species, you'd want to sample broadly. Okay. So I think I've made these points kind of multiple times, but I'll put it a different way. Does have a different way of thinking about it. Is that what's happening in human genetics is we're sampling more broadly. We just are, right? And we just can't, not sample more broadly. So given that we're sampling more broadly, we're going to be discovering lots of variants from different parts of the globe at really low frequencies. So we really need to be thinking about burden tests that pool all that information together, because we're not going to, it's going to be trickier to get many carriers for the rarest stuff. And so unless we get our sample sizes really large. So sample size always wins, but in this kind of sample size for the strongest effect stuff, it's going to be hard. So, using burden test type stuff is going to be really helpful. And then also if there are particular carriers you want to characterize, like particular variants you want to characterize, maybe do follow-up and find the cousins that might all carry that variant, and then use that to do your study. So so that's it. I want to thank Maggie, Marita, Dan, Chris, and Arjun again, and current and past members of my lab and my funding agencies, and put a plug for you to think about coming out to Chicago. We've got a great human genetics community, and that's it. So happy to take questions.
Some questions, comments regarding your burden analysis and the idea of rare variants. So first of all, the idea that narrow populations are the ones leading to the genes that are the most important, causing the most common forms of genetic diseases. That's how we've done it way long before the these biobanks, right? All the relatively common forms of genetic diseases were identified thanks to narrow populations. And I wouldn't give that up so easily. That's one thing. But the other thing is, I think one of the issues with broadening the cohort, if you don't sample enough people from the same population, you're going to have all these rare variants without the capacity to know how common they are in their original population. And so we're going to have a lot of noise. And that's what we've been seeing when we're doing our burden analysis, is that we had to kind of combine the two. So on one hand, trying to enroll participants from as broad populations as possible, but then actually doing the analysis on narrower, smaller populations where we can actually match our cases and controls and have our smaller level population allele frequency.
Mm-hmm.
So we can use, so we will not dilute the allele frequency, as you mentioned.
Mm-hmm.
So you can say, okay, this allele frequency is too high in this subpopulation.
I
But
see
that's
what
why
you're
also
saying, yeah.
NOMAD is offering the subpopulation allele frequency and not just the general allele
Mhm.
frequency. I
Mm-hmm.
think having the narrow population allele frequency is extremely important
I
in
see.
being able to show some causality between rare variants.
Yeah, I see what you're saying. So the point is that in doing a burden test, you'd like to filter and make sure the variant truly is rare locally,
Yes.
like in prioritizing those variants.
Exactly.
Like one of the filters is frequency. And if it's a shallow enough, broad, shallow sample, you don't know if that singleton, doubleton is actually at a pretty reasonable frequency in the local group that you've
Exactly.
looked
Which
at.
was the
Yeah,
shock,
yeah,
right?
yeah,
Because you're
yeah.
showing that if
Yeah.
you are narrowing the breadth of your sampling,
Mm-hmm.
you are going to reduce the number of very rare variants.
Mm-hmm.
Because you are getting more individuals with these super rare variants, and
Yeah.
you're sampling too few people from this population.
Yeah.
We thought it's rare.
Yeah,
but
they drop
actually
out
it's a-
completely. You don't even discover them.
Yeah.
Yeah, yeah, you don't even discover them. And yeah,
So unfortunately, that's
yeah.
a very large cohort that's down.
yeah. That's an interesting dynamic I hadn't thought about. Like the so point well taken. Yeah, thank you.
So I have a question about, so this nice plot, when you sample on the PCA space versus the geographical space, and there's not much difference, but I think it's mostly because UK biobank is very homogeneous, right? So if we were to use this to build an epidemiological study in the modern New York, for example,
Yeah,
I wonder if you do the same study on all of us, I wonder if what you see on the PCA sampling would be very different than what you see on the geographical sampling, because actually the geography does not necessarily
correlate with the
yeah.
And yeah, I think that's totally going to be a factor. Yeah.
That relates to the use of race in genetic studies, right? Because you can't do geographical sampling. You have to know race and ethnicity if you wanna sample enough local sampling of the population, and perhaps it's better to do it on the PCA space without labeling people.
Yeah, I think the latter would be interesting to try, right? Is to sort of be, if the goal, this goes back to sort of the NASEM type, what's our goal? Is it to sample sort of broadly in genetic space, right? Then we should be looking in, a low dimensional representation of that space, and rather than using proxy variables like ethnicity or race, which are quite crude for that purpose. Yeah. But yeah, point also well taken on that, like the uniqueness of the UK Biobank for that comparison. Yeah.
Just want to make sure that
Yeah.
we also have questions from online.
Yeah,
So
I see one
question from Guy Sella.
Yeah, should we let's see, Guy, let me see can you try saying something, Guy? Can you unmute yourself and speak?
Hi, John.
Hey, okay, great.
So I was wondering, the plot you said about the power in terms of width for both GWAS and burden tests are very interesting. In some of them, like for the GWAS, but maybe I don't remember the y-axis, it doesn't appear that you have an increase in power, but it's not enormous. And if I'm getting this right, as you mentioned towards the end, there's all the problems with population structure confounding.
Yeah.
So I'm just wondering, I totally take your point that at the end of the day, people will sample what they're sampling, and at least so far, study design has seemed to play a little role in it, but hopefully the kind of work you're doing would change it. But how would you balance between the two there for GWAS? And then if we move to burden tests, then I'm guessing that at some point, which we might not be very far from, at some biobanks, you might already get to a point where you have good enough power for most genes, for example. So I'm just wondering between this balance with population structure confounding, which could be a real issue,
Yeah.
and could be a real issue for effect size estimation, which
Yeah.
eventually is going to play a central role in portability as well.
Yeah.
Then how would you think about balancing these things?
Yeah. Yeah. Great. So there's a number of cool like premises to those questions. Um, and yeah, so right, you could think about the burden test power, uh, you know, maybe being only limited, the saturation of burden test power, um, and the confounding issue. I'll say that we're preparing the first manuscript, and what we decided to do is just present sort of the effects on variant discovery and not show those power curves, in part because we want to, because that's what we feel confident about talking about. The precisely because we haven't dealt with the effects of confounding and the both kind of statistically and in thinking about, in terms of like adding, degrees of freedom, so to speak. Um, what we're planning to do is a more detailed simulation study where we're actually doing this next step of the GWAS, like subsampling UK biobank at different scales, doing GWASs and asking like, how did it affect our recovery of variants that you see in the whole sample? And we think that'll allow us to get like a more direct view of some of these challenges. So we're sort of getting a lot of that set up. The challenge is with this theoretical framework that it's hard to get at those, to kind of, it's really well set up for getting at the site frequency spectra, but and we can pass it through these basic power functions that depend on frequency and a number of variants. But to really get at the issue of confounding and controlling for it is hard. Could we get at the like saturation of burden test power? I think we probably could actually, within the limitations of this approach. So that's a cool point that I hadn't thought about. So I hope those are some helpful comments.
Thank you. And it's great that somebody's looking at, actually study design considerations about this.
No?
So your theory is at the single variant level, right? All the models that you made. So am I wrong, or in the theory, there should be also an interaction part? It's conceivable that for certain population, the way of decay or decline of the frequency over time is also driven by additional variants that could, if in the population the compensating variant is present, then would allow the deleterious variant to be present only in the population and not in others.
That's right. Yeah. So the theory is all in terms of like independently evolving mutations. And that's motivated by like a statistical fact that when mutations are rare, it's so rare that you're going to see like the damaging variant on the background of another damaging variant or on the background of another, rare compensatory mutation. So that you can effectively ignore those interactions. They're basically saying there's no interactions amongst these rare variants. But if there was sort of a common variant that is a modifier that you know,
That's exactly.
yeah,
Possibility,
Yeah,
right? Because
yeah.
the mechanism of disease for one
yeah,
term of disease, like let's say gene expression
Yeah.
method, we have a activating common variant that is
Yeah.
neutral normally,
Yeah.
that will make all the other variants
yeah.
neutral.
Yeah, I might revert, my argument there would be a lot of these common variants are shared across the globe. They vary in frequency, so maybe it's, it might be 40% in one population and 45% in the other. Then you have to ask, is that difference gonna drive the evolution of a rare variant, through that interaction? So that kind of stuff we're missing, but I kind of think it might be,
There are few that are exceptions.
kind of on the edge, yeah.
It would be really interesting to also see how sampling or empirical analysis of US-based population
Yes,
compares to
yeah.
the UK biobank, where, in the US, obviously, we have migration, admixture, and much more structure in terms
Yeah.
of populations.
Yeah, it'd be
What's
nice to be
your-
doing this in the all of us
Yeah.
context. Yeah. So, right. So that's interesting in terms of like how structured are these particularly rare variants in the US. We know like, they're kind of these anecdotal story of, colleagues, Nelson Freimer used to work in populations in Costa Rica, a lot of founder effects in some of these, due to like European founder lineages bringing in particular mutations. And the time scale basically of variants arising, in founder lineages in the US and then the spread amongst the descendants. But we might expect like these sigma type values to be higher in US history, both because of multiple founder lineages arriving different corners of the US. Also just generational movement in the US maybe being larger. So yeah, my gut would be like the difference between doing a study just in New York, a New York based cohort versus a New York, Denver, San Francisco might have a shallower impact than doing, London, Paris, Berlin or something, Yeah.
Great. I don't see further questions online.
Okay.
So thank you so much. This is
All right.
fascinating.
Thank you.
Thanks, everyone.